All right. And again, as per usual, if anyone's trying to get in, oh, like that, I think I let them in, but just let me know. Okay, so we're gonna start with B cells. I put this on here because I forgot to say something that I wanted to say from yesterday when we were talking about hereditary angioedema. I wanted to mention that so hereditary angioedema can actually mimic symptoms of anaphylactic shock, and we're going to talk about anaphylactic shock, I think, next week. But if you're the physician on call and you think they're having an anaphylactic reaction, and then you try to give like epinephrine to that patient, a lot of the times it's like the swelling's not going to go down. And that's like another indication that it's not like an allergic reaction, it's not anaphylactic shock, it's actually hereditary angioedema. So I wanted to highlight this because on your like objectives from Dr. Milani, she has these drugs on there. She has antihistamines, glucocorticoids, and epinephrine like listed as drugs. So I was trying to think like, oh, maybe that's why she has them on there for like a hereditary angioedema question. You know what I mean? So if you see, and we're going to talk about like what anaphylactic shock looks like, but like if you think the patient's having anaphylactic shock and then they give them like one of these meds, like epinephrine, antihistamines, glucocorticoids, and nothing happens, then it's hereditary angioedema. Okay, so it's just something I wanted to emphasize because I noticed these were drugs that were on your objectives list. Okay, I wanted to include that in the recording too. And I think this was like the last question from yesterday. Okay, so let's talk about B cells. So what we're doing is we're going to start off by talking about antibodies and then go into B cell development and then B cell activation and then end with like a bunch of questions. Feel free to leave like obviously whenever you guys need to. It might be like very long, but the questions are going to be really good at like at least showing you what you should be focusing on because there's going to be a lot of content. Okay. So antibodies. So an antibody, another name for it is an immunoglobulin. So if you know, like, we talked about IgM and IgG yesterday with like activating the classical complement system. IG in like IgM and IgG stands for immunoglobulin. So it's like it's the same thing. We briefly talked about this when we talked about the B cell receptor. But basically, as like a review, it consists of heavy chains and light chains. So the heavy chains are the ones that go all the way down. They're the long ones. And then the light chains are kind of the ones on the side here. These are light chains. There's also variable regions and then constant regions. So the variable region is going to be like the very top. So see like the V, so VH, VL. This stands for variable region of the heavy chain, variable region of the light chain, and then same thing. It would be the same thing on the other side. The constant region is like the C. So constant region of the heavy, constant of the light, et cetera, et cetera. But remember, this whole portion, like this whole upper portion is called the FAB. This is what's binding antigen. And then this bottom part is called the FC. Remember, C does not stand for constant. It stands for like crystallizable. Because the constant, the constant region extends up into the FAB region. We're not going to go too much into these hypervariable regions, but it's just like a fun fact that like within the variable region, there's hypervariable regions, and that just is what we're going to talk about this process today where like that can kind of change to bind an antigen like really, really nicely, but you don't have to know the specifics of that. It's just like a portion of the variable region. So we talked about this. The FAB is what's binding the actual antigen. Specifically, remember we talked about how it's like a paratope binding to an epitope. The epitope is like the exact amino acid sequence on the antigen that the paratope on the antibody recognizes. So it's just that terminology again. More terminology, and this is what they'll try to trick you on questions. The FAB portion, so this portion, this is what determines what's known as the idiotype. So the idiotype is basically like, and we'll get into it a little bit more, but that's the portion that's like the specificity portion. That's the portion that's binding the antigen. That's the idiotype. I'm emphasizing that because down here we have another thing with type in the name, and that's something that people get confused on. So then the FC portion, something that I wanted to mention here is that macrophages bind to the FC portion, remember, and a bunch of other cells do too. Remember we talked about ADCC yesterday with NK cells and with eosinophils, they can participate in ADCC, actually on Tuesday, sorry. But remember like macrophages, NK cells, eosinophils, they have FC receptors on their surface, and it's recognizing the FC portion of the antibody, which is down here. So macrophages have it, neutrophils do, NK cells do, eosinophils do. A lot of cells have FC receptors that recognize this FC region. Also, complement, this is where complement binds. And I think I really wanted to emphasize this because you would think, because remember we talked about how antigen antibody complexes activates complement. That's like your classical pathway, right? When C3b is binding to this antigen antibody complex, it's binding to the antibody. It's binding specifically like down here. And then finally about this FC region, this is what's going to determine what's known as the isotype. So that's why I wanted to point out this terminology. Don't get confused between these two, because you can know the answer and then forget which one's which. So the idiotype up here, that's like the specificity of the antibody. Isotype is like, another name for it is class. We're going to talk about class switching later. The isotype is basically like the class of antibodies. So IgM is one class. IgG is another class. That another name for class is isotype. Sorry, question. Paratope and idiotype the same thing? Thanks. That's a good question. So paratope, I think it like is in theory, but it's like paratope is the exact amino acid sequence that recognizes the epitope, whereas the idiotype is more about like specificity of the overall antibody. So like if we had a mutation that made one of these hypervariable regions less able to bind to an antigen, that would like the idiotype would change. If that makes sense. It's not really like, I guess technically too then the paratope would change, but it's kind of like epitope and paratope are going to go together. Okay, good. I know there's a lot of terminology. And then we talked about this like briefly on Tuesday, but the hinge region, this is very, very rich in prolines. This is your biochem tie-in, and the prolines are kind of what allow it to like shift around if it needs to. And then there's these interchain disulfide bonds. So you can see them here, here's some here, here's some here, and here's some here. So there's always disulfide bonds kind of between these chains holding them together. And then as I kind of mentioned, the paratope on the antibody is recognizing the epitope on the antigen. And this is going to fit in, for the most part, a lock and key kind of fashion. Okay. So there's three main functions of antibodies. So the first one's kind of like intuitive. So it neutralizes. So we have a bacteria, antibodies can just kind of stick to the surface of it, and then it basically can't do stuff. It's neutralizing the function of that bacteria. This also works for like toxins. So if this bacteria were to secrete like a toxin, we don't want the toxin to enter our cells. So antibodies can actually bind to it and stop it from even being able to do whatever it's supposed to do. And that's like neutralization. You're neutralizing the function of like whatever it's binding to. And we'll talk about vaccines next week, but this is kind of what like certain vaccines that you get are trying to do. They're trying to neutralize toxins of certain things like diphtheria, pertussis, what's the other one? DTaP, diphtheria, tetanus, and pertussis. You're trying to block that toxin. You're trying to neutralize it. Another function is opsonization. So remember I said C3b is like the best opsonizer you can ever have in the entire world. There's also, antibodies can also act as opsonizers. And this should hopefully make sense because we just talked about back here that macrophages have FC receptors on their surface. So if there's an antibody bound to a pathogen, it's going to look really tasty for a macrophage to come and kind of engulf it. And then we talked about this yesterday, but we know that if you have antigen antibody complexes, you can activate the classical complement system. And then complement, you're going to get more opsonization, you're going to get lysis, and you're going to get like the recruitment of neutrophils and other inflammatory cells to the area. Okay, so these are the three main functions of antibodies in general. This is kind of what I was just kind of mentioning, going a little bit into a little bit more detail. So neutralization in general, you're just kind of intercepting the antigens before they do anything. So I put here, it's like you're simply inactivating. You're not like actually killing anything. You're just kind of like inactivating it. You're not letting it do what it's trying to do. So an example of this is like antivenom. So if you're bitten by a snake, they don't give you the magic, like they don't give you something that's going to completely get rid of that snake toxin. They just give you antivenom, which is just antibodies that bind up the venom and don't let it do anything. And then eventually this will get cleared by like your spleen, right? By the reticuloendothelial system. So opsonizations, like we said, IgG and C3b are the best opsonins. So this is really, really important for clearance of your encapsulated bacteria. You guys all better know what are these three main encapsulated bacteria that you absolutely need opsonization for in order to get rid of. You can say it to yourselves if you want, but you can also type it to me. I know someone knows. I know someone remembers. Good. H influenza. Good. SHN acronym. Okay, yes. Yeah, Neisseria. Good. Neisseria, strep pneumo. There you go. Beautiful. So we get strep pneumo. I'm not going to type this all out. All right, so strep pneumo, Haemophilus influenza, and Neisseria. And your Neisserias. So those are the three. You need to know these. I will ask it in every single PowerPoint. The reason why is because these three, since they have capsules are really good at like hiding cells from the immune system. So it basically prevents like splenic macrophages from seeing the microbe. So they just can't see it. Think of it as like being slimed. Like, you know, when you go to like Disney or something, get slimed and it's like, you're kind of, you know what I'm saying? So it's kind of like they can't see you because you have like this covering, and that's kind of what a capsule does. Oh, I did put it here. I forgot I put this here. Yeah. So it's like they're covered in slime. So like the immune system's like, I don't really know what that is. And like there's no like antigens that are present. It just kind of is like invisible. So that's kind of why these opsonins are really important, because it's tagging the capsule. It's like, hey, this is ready to go. Okay. So it allows the immune system to see it. So that's why opsonization is really important. Opsonization isn't just for encapsulated bacteria, but it's really, really important to get rid of your encapsulated bacteria. And then finally, complement recruitment. Remember, IgM is the best activator of complement. We're going to talk about why today. But remember, you can also have 2+ IgG molecules. And remember, the mnemonic is GM makes classic cars. So G and M both activate the classical complement system. Okay. And then, of course, like with this, you're getting more opsonins with complement. You're getting the membrane attack complex. So it's just a good thing. Okay, so going into the 5 antibody isotypes. So isotypes determined by the heavy chain. So we have IgG, IgM, IgD, IgE, and IgA. So IgG, this is going to be the one that's going to be present in previous infection, or like if you're considered immune to something. This is like, again, what you're trying to do with vaccines, which we're going to talk about next time. But IgG is the one that's going to tell you that you're immune to something. So I put here, like if I have COVID IgG antibodies, either I had COVID in the past or I was vaccinated against COVID. There's no way to tell because you're going to have IgG. That means you've recovered from the infection. You've recovered from the vaccine. Okay, so that's kind of what you're trying to make with vaccines too. IgM, and this is going to come up a lot. We're going to talk about this, like why this is the case. But IgM is what's going to be present when you have an acute infection. So if I have COVID IgM antibodies, that means right now, currently, I have COVID. So this is like very helpful for when you're doing, like when you're ordering lab tests and it's like you get like these antibody results back and it's like COVID IgM is negative, but COVID IgG is positive. That would mean like, okay, patient doesn't have COVID right now because IgM was negative. And then patient either had COVID in the past and recovered, or patient was vaccinated against COVID and like made an immune response against COVID. And then we'll go into detail with the other guys as well. Okay, so here's IgG. So IgG is a monomer. This is the main antibody produced in the secondary response to infection, which is also known as the anamnestic response. And we're going to talk about that a lot in the vaccine session. But again, this is just kind of think of this as being like you have protection or you have immunity to something. That's like what IgG's, like that's what the role of IgG is. It's most abundant in the serum. It's like 75% of your serum is like 75% of all the antibodies in the blood is IgG. And then some of its roles is to neutralize microbes. It fixes complement and it's a potent opsonizer. So all those things on that slide, when I had those 3 functions of antibodies, IgG can do all of those. And then something specific just to IgG is the only antibody that can cross the placenta. And we're going to talk about why this is like kind of important later. Okay, so IgM is a pentamer. So it's a pentamer and it means it has 5, basically it's made up of 5 IgM monomers. That's what a pentamer is. Okay, so I'm just kind of drawing all the 5 IgMs. And then there's what's known as a J chain, and the J chain is kind of what allows all of these to kind of come together and become a pentamer. This reason, the fact that it's a pentamer, this is why it's the most potent activator of complement. Because remember I said complement is made up of antigen-antibody complexes. Now we can have basically 10 and 5 complexes, but we're going to have like 5 for the price of 1, right? So that's why it's really, really, really good at fixing complement. And this is the first antibody produced in response to an infection. This is why, like, if you have COVID right now or you have an infection right now, you're going to have IgM. You won't have IgG, you'll have IgM because it's like the first. So again, if you're trying to see if someone has like an acute infection, you're going to test for IgM levels because it's the first one produced. And then it has what's known, it's the highest avidity antibody. So I'm going to talk about down here what like avidity is versus affinity. But first, I want to also mention that IgM is the B cell receptor. So that's why it's a monomer. So it technically is a monomer, and then the B cells, when it starts to secrete stuff, which we'll talk about today, it'll start to basically make this pentamer in the blood. So it's found in circulation as a pentamer. It's found on B cells as a monomer. It's the same antibody. Okay, so then affinity and avidity, it's just more terminology. So affinity, you can think of it as how strongly the FAB portion binds to the antigen. Like how strong is that interaction? That's affinity. Avidity is basically a sum of all the affinities. So I thought that this was a nice picture to kind of see, like this might be like an IgG molecule. So you can see its avidity is 2 because it has 2 like spots that it can bind to antigen. IgM is a pentamer, so it's going to have like 10 spots. So it has 10 spots where it can bind to antigen. So that's why it has a very, very high avidity. Does that make sense? Affinity versus avidity. Affinity is really just talking about the strength of this interaction. Each interaction is since there's 2 binding sites for antigen, like, yes, there's 2 technically, but like it's just the strength of this interaction. It's going to be the same on both sides. Whereas avidity is like the sum. So how many interactions are we having? That's avidity. Okay.
Christina?
Yeah.
So for avidity, I just want to double check I get it. So is it like the possibility, like, does it have to be bound, or is it like the possibility of how many things it can bind?
Yeah, good. It's like the possibility, like the potential. So like IgM's avidity is like 10 because it has the potential to bind 10 antigens. But for example, IgM's affinity, I mean, it doesn't really make sense to give it a number, but like the affinity would be 5, even though, again, it doesn't make sense to give it a number. But you see what I'm saying? Like it's like the strength of the interaction of the one antibody in this pentamer. A lot of the times people say you can like avidity is usually twice affinity because there's 2 spots. So it's just like you double it. But I think it's easier to think of it as like the strength is the affinity. Like if you have a high affinity for something, it really likes it. Low affinity, it's like, eh. Whereas avidity is just how many antigens can it bind, basically. Okay, so IgA is found mostly as a dimer in secretions, and this is more clinically relevant, is that IgA is found in secretions. It also has a J chain, so now we have 2 antibodies with J chains. So IgM has a J chain, IgA has a J chain. And when it's secreted, and it's found in those secretions, so think mucous membrane, think your GI tract, think tears, saliva, breast milk, all that stuff. Those are all secretions. It's going to exist as a dimer. However, note here, since it dimerizes in this way, it can't fix complement. There's no exposed FC region, so you can't, IgA does not fix complement, but that's fine. It's not what it's trying to do when it's in the secretions. So fixing complement means it can activate complement easier. Yeah, so if you can fix complement, that means that a C3b molecule can bind to the FC portion, that you're fixing complement at that point. Which I don't know why I drew it there, because a dimer can't fix complement. It's like right here, technically, like C3b could bind to the FC portion here. Here it's not exposed. This cannot fix complement. C3b cannot bind.
So would you say that IgA has a higher avidity than like IgG?
Yes, very
Okay.
good. But you can't say for sure like about the affinity. You have no idea about the affinity because it's dependent on how strong that interaction is between antigen and antibody. Okay. Again, IgA's most important role is to exist as a dimer. It doesn't fix complement. It's fine. We don't need it to fix complement. We need it to kind of protect like our mucosal membranes. So again, tears, saliva, breast milk. I highlighted this for a reason. the GI mucosa, so like those pyre patches, malt tissue, which is just another type of mucosal tissue. And IgA is what's going to be primarily protective against GI infections such as Giardia lamblia. And I put the buzzword B here because Giardia, if you have one of the immunodeficiencies we're going to talk about next week is selective IgA deficiency, where patients just don't make IgA, and you're going to see like they get so much Giardia infections because Giardia, how it infects you is it comes in through the GI tract. So you don't have an antibody basically where the GI tract is at that barrier. So you're kind of allowing for Giardia to easily enter. Does that make sense? So we'll talk about this more with the immunodeficiencies, but this is going to be a buzzword. If you see like recurrent Giardia infections, that's an IgA deficiency. However, when it's like circulating around in the blood, it's a monomer. So here's IgA as a monomer. It's actually the antibody produced in the highest amounts, but it's produced in the highest amounts via the secretions. So it's not the highest amount in the blood, that's IgG. IgG is very, very present in the blood. IgA is the one overall produced the most, like the highest, but most of it is found in the secretions. In the blood, I think it's only like 10 or 15% overall, something like that. Probably 10. And then basically how it undergoes this, so it's going to circulate like this through the blood like this, and then in order to kind of become a dimer, it undergoes this process known as transcytosis, and it picks up a secretory component. And I'm going to show you what that looks like. And I only mention this because there is a genetic defect where this can't happen. So again, this is kind of like your selective IgA deficiency, right? So what happens, here's IgA and here's your dimer. So we're in the tissue. So now we're like in like a mucosal like area. There's a certain receptor, it's called poly-Ig receptor. You can also see it as like PIG, like pig, like that, poly-Ig receptor, that binds to IgA. And then it's going to undergo a process known as transcytosis, which is just taking it from one part of the cell to the other part of the cell. And then when it releases it, something known as the secretory component, which is just a part of the poly-Ig receptor, remains bound. And this is now what allows it to be secreted. So again, you have a genetic defect, maybe in the poly-Ig receptor. It's going to look like an IgA deficiency because now you're not going to be able to secrete IgA. Okay, so IgE, this is the guy that's going to bind to mast cells and bind to basophils. So remember how we talked about Tuesday with ADCC, how, well, I guess eosinophils too. I'm going to put, it's down here. Eosinophils also have Fc epsilon receptors. So remember I said like if you have a cell and it's like Fc whatever receptor, that's the type of antibody it can bind. So this would mean it like this eosinophil has an Fc receptor, like it can bind, it's a receptor for the IgE antibody. So here's its receptor and it can bind to IgE. So this would be called the Fc epsilon receptor because this is IgE, E for epsilon. Okay, so mast cells and basophils also have Fc epsilon receptors. And we're going to talk about this with hypersensitivities next week, but this is kind of what's mediating your type 1 hypersensitivity. Type 1 hypersensitivity is basically just allergies, like seasonal allergies. If you know anyone that has seasonal allergies, they have tons of mast cell degranulation, tons of basophil degranulation, tons of IgE present. So what happens, and I like this picture here, when you're exposed to an allergen, so like I said, mast cells, basophils have IgE on their surface. Like it literally has IgE on their surface. When you're exposed to an allergen, like right here, it's going to cross-link the receptors, and you really need this cross-linking. This is like kind of what triggers it. If like a random antigen bound over here, it's not going to work. You need cross-linking of at least 2 IgE molecules. And then this cross-linking, it's kind of a trigger for the mast cell, hey, degranulate, release histamine. If this was a basophil, like release your mediator, right? We talked about the mediator releasing cells. Okay, so mast cells, its mediator is histamine. So this is why you have to take an antihistamine, something like Zyrtec or Claritin or whatever. It's trying to block this histamine. It's actually an antibody. It's actually a neutralizing antibody. against histamine. That's what antihistamines are. Okay. And then, so the other thing that it's like IgE is involved in is parasitic infections. So specifically with helminths. So helminths are like the really long worms that I showed you the image of on Tuesday. And eosinophils also have Fc epsilon receptors, right? So they're going to have the epsilon receptors. So what can happen is, well, this is the worm, sorry. So IgE is like, oh, there's a worm here, we have to get rid of it. So it's going to bind it. Remember, with its FAB portion, that's the portion that's binding antigen. So it's binding it. And then here come eosinophils with their Fc epsilon receptors, and they can bind to the Fc portions of IgE because they have Fc epsilon receptors. And then once it's bound like that, all these eosinophils bind, that's a trigger for its mediators to get released. And they're trying to destroy this helminth. And that's just another example of the ADCC, the antibody-dependent cellular cytotoxicity. Okay, so if you see eosinophilia when you order a blood smear, so if you have a blood smear and you see the eosinophil levels are really, really high, think parasitic infection, because that's what's basically drawing all these eosinophils over to, like, out into the blood. Good? Everyone with me? Okay. Do basophils work for parasites too? I guess in theory they could, but basophils are more so with allergies. And same with mast cells. Mast cells and basophils are more so with allergies versus eosinophils, which are more so for like parasitic infections. But I guess in theory they could. I guess it's just something that they just don't do. Okay. So IgD, the greatest mystery in immunology is what IgD does. No one knows what IgD, it literally has no role. Like, they don't know what it does. They're like, it must have a function, but they have no idea. And I know that this is a meme, but this is actually a pretty good, like, summary of each of the other antibodies. So I think it's great. But yeah, IgD, no one knows what it does. But we're going to talk about B cell development today. IgD is found on your mature B cell surface, and this is actually how you can determine you have a mature B cell, is if you have both IgM on the surface, like a B cell receptor, right? And IgD. It's just like a marker that you have a mature B cell, even though it literally doesn't do anything. And we'll talk about that today. But that's just like a way that you can tell you have a mature B cell. So I guess it's like, its function is it's like a marker of a mature B cell. Okay. So those were antibodies. So now we're going to jump into development of antibody diversity. So we have to kind of make very diverse antibodies in order to see all these different antigens that we're like seeing on a day-to-day basis. So there's two types of like this development process. One is antigen independent and one is antigen dependent. And it's like, just kind of think of what this phrase means. So antigen independent means that you're getting diversity before the B cells, before the antibodies even seeing antigen. So it must be something like genetic, like happening on like before the B cell, before the antibody even like goes out into the peripheral blood, which is exactly what's happening. It's a bunch of like genetic things. We're going to dive into it, but it's a lot of like generation of the actual antibody, like the heavy chain generation, the light chain generation, all this different stuff. There's all these different kinds of like processes that can occur to help give you like a very diverse antibody from the start, before it even sees any antigen. So I think of it as like having like, you're developing all these antibodies with this inherent diversity. So like you should, in theory, be every single antibody you develop should look different to start. And then once it's like you made your antibody, you put it on the surface of a B cell, you're letting the B cell go to the peripheral blood to go find some pathogens. Then it becomes then you have an antigen dependent process. So now, like, once these receptors are going to bind to antigen, there's a different process that can occur now that's going to make this antibody even more diverse. And that's where you get more of like your lock and key. And these two processes are known as somatic hypermutation. Another name for it is affinity maturation. And then isotype switching or class switching. And we're going to talk about all of these. It's just kind of like an outline. I think this is the coolest word ever, somatic hypermutation. I just think it's so cool. But it's basically just like making this, once it sees antigen, it's making it like super, super, super specific for that specific antigen. That's all that somatic hypermutation is. And then isotype switching is like, let's say we need more, I don't know, IgE. You can switch from one class of antibody to another. And again, we'll talk about that. Okay, so this is kind of the outline of what we're going to go into. So let's talk about B cell development. So there's two like types, like as I just kind of mentioned, there's antibody independent and antibody dependent like development of an antibody. So centrally, so remember where B cells are developed, bone marrow, B for bone marrow for B cell. This is where you're going to get all of those processes occurring that I just kind of mentioned before it even sees an antigen. So VDJ rearrangement, there's an enzyme known as TDT. It can make errors, which can actually lead to diversity. And then positive selection. So we'll talk about what positive selection is. Then we have our B cell precursors are going to go to immature B cells. And then we have a process known as negative selection. Again, we're going to talk about it. And if it passes negative selection, then it can leave. It can leave and go see if it can go find antigen. So it's going to go to some sort of secondary lymphoid tissue. Remember, secondary lymphoid tissue, spleen, lymph nodes, Peyer's patches, anything that like is basically like lymphoid in nature that we talked about on Monday. So if a cell successfully passes negative selection, now it can kind of finish undergoing its maturing process. And by this, I just mean like now you can go through antigen dependent development. So it's going to be exposed to an antigen, and then we go through those processes we just talked about: class switching, somatic hypermutation, or affinity maturation. So again, just kind of like another outline. And I wanted to bring up the words here. So you're going to see like these things today and tomorrow with T cell stuff. Central and peripheral, like there's this concept known as central and peripheral tolerance. Central, they're talking about like the primary organs. So central tolerance for a B cell would be like we'll talk about tolerance, but things happening in the central organs, which are your primary lymphoid organs. Peripheral tolerance, or anything with this word peripheral, means now it's in the peripheral blood. So it's in like a secondary lymphoid tissue or something like that. So I just kind of wanted, if I'm going to start throwing these words around, and I want you to kind of keep that straight. So central, we're talking about like the bone marrow before it even leaves. And peripheral is going to be, okay, it left, now it's circulating around. Sorry, question. So does B cell technically go through both of the developments? Yes. So the B cell goes through all of this. The B cell is going to go through like central development and peripheral development every single time. It's going through positive selection and negative selection. It's going through all of this stuff. Okay. So tolerance, again, we're going to talk about tolerance a lot more tomorrow. T cells are like, because T cells are like the ones that can actually like, these are kind of the ones responsible for autoimmunity. B cells can also be responsible for autoimmunity, but T cells are like the ones that are really, really responsible for it. So heavily tested is like T cell, like the process of T cell development, and these like this idea of tolerance. So central and peripheral tolerance, again, central, bone marrow, peripheral, we're in the peripheral blood, we're out of the bone marrow, we're in those secondary lymphoid organs. Central tolerance is basically when you're removing any like B or T cell that doesn't pass this process of negative and positive selection, which I know we haven't talked about yet, but just keep that in mind. It has to basically pass these checkpoints to make sure like, hey, are you reacting with self-antigen? If you are, you can't leave. That's kind of what like these selection processes are. And if it doesn't pass, they're removed. So it's like you have removal of these self-reactive B and T cells within the primary lymphoid organs, because again, we're in central tolerance. So it's not, these cells aren't even allowed to leave. That's basically central tolerance. Peripheral tolerance is the same idea, but you're kind of, it's like a fail-safe mechanism. Like, hey, what if some of these self-reactive B and T cells left? What if they did leave? Then the body has like a second mechanism called peripheral tolerance to kind of catch those guys, catch the escapees. So peripheral tolerance is when you're going to remove the cells that escaped central tolerance. So it's the same thing. You're removing self-reactive B and T cells, but now we're outside of the primary lymphoid organs. We're in the peripheral, we're in the secondary lymphoid organs. And I'm emphasizing this now because then it'll make tomorrow a lot easier. And it's a little bit more complicated with T cells. So at least if you really grasp it now, or at least just have an idea of what I'm talking about, it'll make tomorrow a lot easier. Okay. So B cell precursors. So a lot of the times, like they will ask these like random, because they want to see if you understand B cell development. So they will ask these like really random questions about like, do you know what's on the surface of all of these cells? which is kind of annoying. But what's going to happen, and I'm going to show you like the kind of the process step by step. But we start with a stem cell, right? We start with like a stem cell that's going to turn into a lymphoblast, right? And then we're going to go through that development process. We have heavy chain genes, which are here, and light chain genes. So again, talking about the antibody. So here's our antibody. Oops. I'm going to start over. So here's your heavy chains, and then here are your light chains. The process of like heavy chain, like diversity and light chain diversity is kind of separate. But so like you have to kind of consider the genes to do all of these, like you kind of have to consider the genes separately. That's why there's these two lines. But remember, heavy chain is referring to, is it IgM, G, D, A, or E? Whereas light chain, it's either going to be kappa or a delta. I don't think I mentioned that, I'm sorry, on the slide of the antibody. Any antibody is going to have kappa or delta light chains, and it doesn't matter, they do the exact same thing. Whereas you saw like all of the heavy chain antibodies do different things. Light chains, they're interchangeable, kappa, delta. Just kind of recognize kappa or delta as being like the light chains. Okay, so I'm going to go through this slide, and if you're like very confused, I'm going to put this in pictures because I think it's a lot easier to kind of see. But once we have a stem cell, nothing's really happening right now. And then we start to go through the next stage of B cell development where we have like an early pro-B cell. So it's called a pro-B cell. So a pro-B cell you're rearranging certain genes within the heavy chains and within the light chains. Okay, so you're rearranging certain genes. I know it doesn't make sense yet. And in order to do this, you need these basically these enzymes called RAG enzymes, RAG. And I mention this because another immunodeficiency is due to RAG deficiency. So I want you to kind of just be aware of what RAG is. So I don't just say, oh, this immunodeficiency is due to RAG deficiency. And you're like, what is RAG? So RAG is basically just what's responsible for doing this rearrangement that we're going to talk about. So the heavy chains get rearranged, then the light chains are going to get rearranged. They're going to start to get rearranged. And then we have our fully rearranged antibody, and we have our immature B cell that can now go and see if it can pass positive and negative selection. Okay. So the steps are pro-B cell, pre-B cell, and then immature B cell. So some notes before I kind of show you what's happening is the heavy chain rearrangement happens before light chain. Rearrangement can only happen in the bone marrow. So again, that's why I kind of like back here when I go back to this slide, we're still in the bone marrow here. This is it's a genetic thing. You can't change genetics really when you're out. Like B cells, when the genetics are changing, it has to be like when they're still developing. So it's kind of like this rearrangement process can only happen in the bone marrow, which requires a DNA polymerase known as TDT, and then these RAG enzymes, which require turning on of RAG genes. Okay, so bear with me. We'll go through the process and hopefully it'll make more sense.
I have a quick question, Christine.
Yeah.
Are RAG 1 and 2 turned off in the late pre-B cell phase so that like the heavy chain doesn't keep rearranging?
So good question, but they're still on. So anytime you see the word rearrangement, RAG has to be on. So
Okay.
this is rearrangement. It's like the process of rearrangement is occurring. RAG has to be turned on.
Oh, okay.
See what I'm saying? It's rearranged. So it's rearranged. It's done. RAG turns off. But now we got to rearrange the light chains. So now RAG is turned back on.
Okay, that makes sense.
Again, hopefully it's going to make sense here. Don't take notes. You don't have to know the specifics of this at all. I just kind of want you guys to see like this process because it doesn't really make sense without visualizing it. So I kind of made like this little diagram of what this VDJ rearrangement is. And we're going to be in the early pro-B cell. So if we go back, we're going to start here. We're going to start with that heavy chain rearrangement first. So the first thing that happens, and you guys can follow along too, but the first thing that's going to happen is this DJ rearrangement. So we have about 50 V genes. We have about 25 D genes and about 6 J genes. So imagine all like 1 through 50 here, imagine 1 through 25 here, and then 1 through 6 here. And then the C genes, these are our constant region, right? So we only have mu, delta, gamma, alpha, and epsilon. So these are the constant region guys. So the first thing that happens is DJ rearrangement. Oh, I forgot I did this. Okay. So this is the variable region possibilities versus the heavy chain constant region possibilities. Okay. That's just what on a B cell DNA, this is what all of these things mean. V, D, and J are going to contribute to the variable region. So the very, very top parts of the antibodies. Remember we have the constant Remember we have the constant regions and the variable regions. The VDJ rearrangement is kind of what's allowing for that variable regions. And again, just think about it. We're trying to generate diversity. Like that's what we're trying to do before it even sees antigen. So it makes sense that we're trying to like make these as diverse as possible before we even see an antigen. So that's what the whole idea behind VDJ rearrangement is. Okay. So variable region, constant region. Okay. So the first thing that happens is DJ rearrangement. So let's just say, let's randomly select D12. I don't know. We just picked a random one and J3. So we picked D12, which is somewhere in here, and then J3, which is somewhere in here. Everything in between D12 to J3, like everything between those guys gets completely spliced out. It just gets excised. So now instead of having 1 through 50, 1 through 25, and then 1 through 6, now we have 1 through 50, and then we only have one D gene now, and then one J gene. That's DJ rearrangement. So then we're going to randomly select a V gene because now we need VDJ, right? So we were in the early pro B cell here. Now we're in the late pro B cell. So if we go back here, we had DJ, that's done. Now we have VDJ. So we're in our late pro B cell. Oops. So let's randomly select a random V. So we got V38. So here's V38. So now we have these three different things. We have V38, D12, J3. And then this is going to be on a B cell, right? And remember, all B cells have B cell receptor is mu, so IgM. So this is going to get, I shouldn't have put an X here because we are going to have D. Remember I said like the mature B cell has D on its surface. So we can un-X this. But mu is the most important one, obviously. So then we have this mRNA of very different from the DNA. And then from here, we're going to kind of keep going. This is actually something that Dr. Demarest last year wanted me to go over, so don't worry too much about this. But I wanted to show you when you're in a large pre-B cell, there's like a checkpoint that happens, and I tried to line up the colors. So we made VDJ, right? So that's in this variable region. I circled it in this variable, within this, what we're making right now. The variable region is right here. So here's our red with the V, here's our yellow with the D, here's the J, same thing, V, D, J, and then the constant region is the rest, what's shown in pink. And what the B cell does is it kind of pairs what we just made. It just kind of pairs this with what's known as a surrogate light chain. You don't have to know these names. She wanted them to, but you don't have to. They kind of pair it with what's known as a surrogate light chain. This is kind of like a quality control step. If the body's pairing what we just made with the surrogate light chains, that means we did VDJ recombination correctly. Okay. So we're in a large pre-B cell. What I kind of wrote down here, if now it's able to divide with these surrogate light chains, now we can undergo VJ rearrangement for the light chains. Okay. So here's kind of what I said. I knew it was going to be kind of confusing. So VDJ is for the heavy chains. VJ is for the light chains. So in our early pro-B cell, it's going to happen in the bone marrow. All of this is really happening in the bone marrow. And we have DJ rearrangement first, and this is of the heavy chains. If this works, and you can see here, it's positively selected for. If this process works, it's allowed to go to the next step. That's kind of like positive selection. From here, with the late pro-B cell, the DJ rearranged, but now we're going to rearrange VDJ. So we undergo VDJ rearrangement, late pro-B cell. If this works, we're going to move on, positive selection. We're going to turn into a large pre-B cell. So VDJ is done. You couple it to that surrogate light chain. This is the one that's undergoing proliferation. So this is going to start dividing. And this is actually why let me go back to this slide. that's why I kind of circled this in red, because this is the one that's going to undergo proliferation. And again, you saw like it passed these 2 positive selection steps. So the body's like really sure that it actually wants to like start proliferating. So this starts proliferating, then we have a small pre-B cell, and now we can start rearranging the light chains. And it's kind of the same process as VDJ, but it's only with V and J. There's no D genes with the light chains. So with kappa and lambda, like if you looked at kappa and lambda DNA, like they don't have the D genes don't undergo VDJ rearrangement. Okay, so this is like a simplified version of what we just kind of talked about. And then here's kind of, I wanted to introduce like the positive selection aspect. I know this is kind of like a weird looking chart, but early pro-B cell, we're getting the heavy chain rearrangement, and first it's DJ, right? So if DJ does this correctly, we're positively selecting. So we're positively selecting to go to the next step. Then we have our late pro-B cell. Now we have VDJ rearrangement. And if this works, we're positively selecting to go to the next step. And then I want to note here, this is not negative selection. This is just not positive selection, which I know sounds really weird. But the default for all of these cells is to die. So it's almost like you have to select for them to keep going. So if you don't get a positive rearrangement, it's not like when I say the word selection, it's like an active process. Like I'm doing something. I'm selecting for it. And with negative selection, that's what you're doing. You're selecting to kill something. Positive selection, I'm selecting to keep it. But this is not negative. This is just not positive. So if I don't select for it, it's just going to die because that's the default. Yeah.
Sorry, I have a quick question.
Yeah.
So for the heavy chain, like, you're, let's say that you have a B cell that selects V12 for its heavy chain. When it then goes to the light chain stage, will it also select V12 for that?
Good question.
Or
Good question.
Uncoupled?
Nope.
Maybe like different numbers.
Yep, whatever it wants.
Okay.
That's why you're able to get like such insane like diversity without even seeing an antigen.
Okay.
Yeah, it's a good question. Yeah, so it can do whatever. It's like 2 kind of separate processes. So then we have our large pre-B cell. This is the guy that's going to start proliferating. We gotta, so the only thing that's not on here is it's coupled to like the surrogate light chain, right? And then the light chain's going to start rearranging. It says like, oh, kappa happens first, then lambda. It doesn't matter. Just light chains get rearranged. But you can see if they're not positively selected for, their just default is to die. So they die via apoptosis. But if they're positively selected for, then we're going to get our immature B cell. Remember, it has IgM, and I don't know why it doesn't show oh yeah, it just has IgM on its surface. And rearrangement ceases. So you can even write here like RAG genes are off. Here, RAG genes are on, RAG genes are on. Like just kind of keep that in mind because it'll make a lot more sense when we talk about that immunodeficiency where RAG genes are not working. Okay. Okay. So from here, here's our heavy chain, here's our light chains. So now we have our immature B cell. It has IgM on its surface. That's what we just kind of made. So from here, IgM on its surface, before it can leave to go to the secondary lymph organs, now it needs to pass negative selection. So I should have put this little flow chart in here, but this is kind of what we just talked about. We just kind of talked about this. So at this point, when we're going through VDJ rearrangement, we undergo positive selection. We're positively selecting for the B cell to continue on. If we don't select for it, it's not negative selection, it just dies. That's the default pathway, is to die. And then once we have our immature B cell, now we're going to go through negative selection. That's what we're going to talk about. Okay. And again, like I just kind of mentioned, when you see this word selection, it has to be an active process. So cells that recognize self-antigen are deleted. This is an active process. You're actively selecting to kill the cell. You're not just letting it do it by default. That's not negative selection. Okay, so here's all of the things that can happen with negative selection. So here's our immature B cell with IgM on its surface, right? if, let's say there's no, so here are some self-antigens. Let's say this B cell does not bind to any of these self-antigens. That means it passed. Yay. So it passed negative selection. There's no reaction with self-antigens. So it's allowed to leave. It graduated. It can go to college. So here it can now migrate to secondary lymphoid organs and then continue the next step, which is the antigen-dependent diversity. So that's one thing that can happen. What happens if my B cell binds super, super strongly to like basically what's known as a multivalent self-antigen? So it's binding to multiple self-antigens at once. It either dies, so apoptosis, and again, this is negative selection. So this is not default. This is like the body's like, no, you're dying. Like I'm selecting you to die. Or what can happen is you can try and rescue. The body actually tries to rescue this B cell. It tries to rescue it. It's like, hey, like, let's give it another shot. We're still in the bone marrow, so we can still go back. We can still go rearrange some more V, Ds, and Js. Let's just go back. This is called receptor editing. I'll put it here. I don't know why I didn't write it. It's called receptor editing. So, and again, you're editing the receptor, right? Because it bound to self-reactive antigens very, very strongly. Like it bound very strongly. So it's like, all right, something's wrong here. But when you do this, when you go through receptor editing, and this is kind of what I was trying to emphasize earlier, the RAG genes are going to get turned back on. So RAG genes can get turned back on. You can go through this whole process of VDJ again to try and make a better immature B cell that doesn't recognize self-antigen. And the body can technically do this as much as it wants, and that's why I kind of put this arrow, so then we go back to the immature B cell. It can kind of go through this process like a bunch of times, but like I feel like it gets to a point where it's just like, all right, we're just going to go through apoptosis. I don't think there's like a trigger that's like, all right, we did it 10 times, that's enough. It's just like sometimes it tries to receptor edit over and over and over, and sometimes it's like, this isn't worth it. What's interesting is if the B cell receptor, the B cell and the B cell receptor reacts to what's known as a monovalent self-antigen. So notice it's like kind of binding, but it's like not both FAB regions are binding. It's like it looks like it's kind of binding to it. It becomes what's known as anergic. Anergy is a really weird concept. It's like you don't kill the B cell, but it can never be activated. So it can never bind to antigen ever again. I'm actually not really sure why anergy is a thing. But so it becomes anergic and then it's allowed to leave. So it's actually allowed to leave and go to the secondary lymphoid organs, but then when it gets there, it actually ends up dying pretty fast. So again, I don't know what the point of anergy is. Could undergoing all this apoptosis cause inflammation in the body? Very good question. You guys haven't had Pathoma yet. Pathoma chapters 1 through 3. He talks about how apoptosis does not cause inflammation. It's actually one of the characteristics of apoptosis. It's a very clean process versus necrosis, who think about like a heart attack. A heart attack causes necrosis. That's tons and tons and tons of inflammation. Dr. Satar in Pathoma, he calls apoptosis cell suicide, and he calls necrosis cell murder. So cell suicide, it's like thought out. It can sometimes be very clean. This is like how he describes it. Versus necrosis, which is like murder, like you're just you're shooting someone, you're sprinting away, right? That's like how he describes it. So apoptosis actually causes no inflammation. Fun fact. If the mature B cell has to have IgM, why are there 5 C genes to choose from? Great question. We're gonna talk about that. That's gonna play a role with class switching, which we talk about later. We're gonna talk about that with like the antigen dependent diversity. Yeah, very good question. So it usually always, like B cells, they always do M first, and then D is the one right after it. So that's the one that gets put on the mature B cell. But yeah, why are A, E, why are those other ones there? And it's because of class switching, which we'll talk about. Self-antigens free floating, are they presented? Another really good question. They're presented. There's actually, I don't remember if I talk about it here. I forget which PowerPoint I talk about it, but they're going to be actually presented to the B cell. They're not just like free floating. And that's actually going to be like an autoimmune disease, actually, if you have, so it's what's known as AIR. So it's called AIR. It's like a complex, and it takes self-antigen and it presents it to B cells. And again, I have this in a PowerPoint somewhere. I don't know if it's in this one. And some autoimmune diseases have a deficiency of AIR. So you're not presenting self-antigens to B cells. And that's why you can get autoimmunity. Now this can escape. Yeah, any more questions? Because it'll just keep building on this concept. So again, the whole point of this though is like, we want to generate diversity, and that's why you don't really have to know VDJ like in too much detail. I just wanted you to see what it is, because like you want to generate as much diversity to see as many possible self-antigens as possible. And air is going to just keep giving it to it like, hey, do you recognize this one? No, cool. Hey, how about this one? So you want to have like this vast repertoire of like B cell receptors that can recognize a vast repertoire of self-antigens. So hopefully it doesn't react with any of them, and then it can leave. So this is even before it comes in contact with like any foreign antigen, I guess I should say, because negative selection involves self-antigen, right? But any foreign antigen, this is how you're generating diversity.
Christina, I have a question.
Yeah.
If you have something wrong with air, like, would the cell like not go through this like selection process?
So it
Like,
still
would
would. It would. When it gets to this step, oh, I see. Like, would it negative selection? Right. Yes, exactly. Like, it wouldn't, nothing would happen. Like, it would just be here and then it would just go. Yeah, I see what you're saying. Yeah, so it like, it can't go through negative selection, right? There's nothing like presenting self-antigen to it. Okay. All right. Yes. See? Yes, VDJ. All right. So then I kind of just wanted to show you a little bit more about energy, because energy is like such a weird concept. So again, like I went back here. So energic is when you have a monovalent self-antigen that is presented to the B cell and it recognizes it. So we have this soluble univalent self-antigen that does not cross it doesn't cross-link the B cell receptor. So it looks like this. It's just like you're only binding the one FAB portion right here. If this happens, it's like, yeah, it's recognizing antigen, but like not super strongly. So we're just going to undergo energy. So it undergoes energy. And then from here, this cell is just completely unresponsive to anything else. It can't do anything. It doesn't get activated. It just like exists as an energetic cell. It goes to the peripheral like system, but then it just like dies. It just dies. Again, no clue why energy is a thing. But that's what it is. It's basically like when it's reacting to like some self-antigen. Okay. Here's everything we just kind of talked about, just all in ones. So I would definitely know at least the stages. I have seen boards questions, like practice questions, where they'll be like you have to know this order. So early pro-B, late pro-B, large pre-B, small pre-B, and then immature, and then mature. And then what kind of makes it up. So, and this is why it's like, don't really like try and memorize, just be like, okay, if D and J are rearranging, RAG's going to be turned on. If nothing's happening, like right here, large pre-B, RAG's going to be turned off here because nothing is being actively rearranged. Then our small pre-B, this is the guy that's going to proliferate, and like that's why there's no rearrangement occurring, because this guy's going to proliferate. Remember, he's got like the surrogate light chain on its surface. Now we have our small pre-B. Now we do light chain rearrangement. And this is like, I highlighted this because I have seen questions on this, and it's like a weird thing that they ask about. I don't know why they do this, but like the small pre-B cell, the mu is in the cytoplasm. And then when it's immature B cell, it's on the surface. It's like a weird thing. So it's in the cytoplasm. I just think of it as like, okay, it's not on the surface. It has to get to the surface, so it's in the cytoplasm. And then remember, your mature B cell has both IgD and IgM on its surface. Okay. So that was VDJ. Isn't there a rearrangement happening during large pre-B? What rearrangement's happening during large pre-B? There's no rearrangement happening during large pre-B. This is when like we have VDJ rearranged, we have our heavy chains completely rearranged, and then the B cell tacks on a surrogate light chain, and then it just undergoes proliferation, and then the small pre-B undergoes VJ rearrangement. Does that make sense? So the early pre-B, DJ gets rearranged. Late pro-B, did I say pre? Early pro-B, DJ gets rearranged. Late pro-B, VDJ gets rearranged. And then heavy chain rearrangement's done. So it's done. It tacks on this surrogate light chain. It's like a checkpoint. It's like, okay, like if you have a surrogate light chain, that means VDJ was good. Like we went through the positive selection here. Like this worked. We went through positive selection here. So now we can start to proliferate a little bit. Undergoes proliferation. And now let's rearrange the light chains. And then we also have like more, positive selection. Like did the light chains rearrange correctly? And if it did, good. So this is rearranged. This is rearranged. Now we're going to become like a mature B cell. Oh, good. Thank you. I know it's a lot. I'm like, it's like hard, like pre-B, pro-B. I'm like, oh my God. So if I say something wrong, I apologize. Just go off of the chart. But I do just kind of wanted to, I was looking through some boards practice questions too, and they do kind of emphasize like knowing some of these characteristics of these B cells. And I think the reason why is because certain cancers, right? Like if we have certain cancers like ALS, like we have a B cell lymphoma or something like that. Did I say ALS? Oh my God, AML. If we have like a B cell lymphoma, it's going to be an immature B cell. So it's like if you know more about the characteristics of what the immature B cell is, like one of these, then you might be able to treat the cancer better. So I think that's why they focus on it. But the specifics of VDJ is not to be memorized. Yeah, so all RAG is anytime you see rearrangement, you need RAG. That's like the best way to explain it. It's a gene and it codes for an enzyme. They're called RAG enzymes, RAG1 and RAG2. So if I want to go through any of these rearrangements, heavy chain, light chain, any single time you see rearranging ing on this chart, RAG is going to be turned on. These RAG genes are going to be on. And then here they'd be off because we don't have anything actively rearranging. Here it'd be off. And then let's say we want to go through receptor editing. It binds a multivalent self-antigen. We want to try and receptor edit. We would have to turn RAG genes back on. And the B cell can do that. It can turn it back on and kind of go back through this process. You're welcome. All right. So the non-self-reactive immature B cell leaves the bone marrow. So how does it know where to go? You guys should know, chemokines. Okay, so there are a bunch of different cells that are secreting chemokines. I think these were on the objectives last year, like for Dr. Demarest, so I would not worry about these exact names or even these cell types, to be honest. Like, don't even worry about these cell types. Just know that there's cells like out in the peripheral, like the peripheral organs, like the secondary lymphoid tissue, that's releasing chemokines, being like, hey, come here. Like I have some cool antigens I want you to check out. So that's how it kind of knows where to go once it leaves. So, and these are cells that are usually found in the secondary lymphoid organs. Like for example, follicular dendritic cells, these are like located in the lymph node and they don't leave. These are not normal dendritic cells. They're called FDCs. These guys they only like exist in the secondary lymphoid, like the lymph node, and they secrete chemokines to get B cells to come to it. Stromal cells, same thing. They're in the secondary lymphoid tissue. Myeloid dendritic cells, these are also kind of, these are a little bit different, but like this is like a professional. Remember I talked about how like dendritic cells are like very good antigen presenting cells. So these dendritic cells are basically going to, if they have antigen and they want to find a B cell, they're obviously going to secrete cytokine or chemokines, right? To try and get the B cell to go to it. So don't worry about these names. Again, I should have taken these off. Dr. Demarest had them on her sheet last year. And again, same thing. The cytokines. No. These were just on the objectives list from last year. Don't worry about these. But what I just want you to kind of see is like, just kind of see the overall thing that's happening. So remember like when B cells or any cells enter the lymph node, they're going to enter through the high endothelial venule, right? That's like how they enter, which puts them where? The paracortex, right? So remember the high endothelial venule, it's going to open up into the paracortex. But these chemokines are what's attracting the B cells to this lymph node. So this would be like a stromal cell, this orange cell, this is a stromal cell. This yellow one, this is a myeloid dendritic cell. Over here's a follicular dendritic cell. So you see they're all located within this lymph node and they're all just trying to secrete cytokines to get B cells to come to it. And then these interactions are what drive the maturations. We haven't talked about this, but when they're saying drives the maturation, they're talking about that antigen-dependent B cell maturation. Don't worry about BAFF and APRIL. We don't care. But it's going to basically secrete these factors to allow the B cell to completely mature, which we're going to talk about. And then from here, now we have our beautiful mature B cell with both IgM and IgD on its surface. And from here, there's 3 fates. 1, it can just circulate forever and ever and ever. Not really. It does have a half-life. Actually, I think it was on this other slide. Yeah, 40 days. So not 5 ever, actually 40 days. So 40 days. But it can just circulate. Like if you never come in contact with an antigen, it'll just circulate like until it dies. But then let's say it does come in contact with an antigen, then it would become an effector cell. So the effector cells for B cells are plasma cells and memory cells. T cell effector cells are different. So this is specific to B cells, plasma cell and memory cell. So this is kind of like what I just kind of mentioned, but like we're drawing the anatomy back into it. So remember the B cell is going to enter through the high endothelial venule. This is a lymph node. So it's going to enter through the high endothelial venule. We're in the paracortex. Remember the T cell zone. So this is your paracortex. And then from here, like depending on the cytokines that are being released, it's going to be recruited. It can be recruited here. It can be recruited here. It just, again, it depends on what cytokine or chemokines, are being released to kind of either activate it or just kind of push it through the circulation. And then it's going to leave and leave through the efferent vessel. Good, so it's kind of like a less busy version of this slide. Okay, so we talked about this. This is happening in the bone marrow, so this is centrally happening. We get VDJ rearrangement. It's allowed to leave if it doesn't interact with any self-antigens. It's allowed to leave. So here it's leaving, and then it's going to kind of undergo maturity as it's just out there. It just kind of becomes mature. This is not too important, this process, but you go from immature to mature. So you go from having IgM on the surface to both IgM and IgD. You don't have to know much else about that. Now, if it finds an antigen, so all these cells are secreting chemokines, the stromal cells, the myeloid dendritic cells, follicular dendritic cells, they're secreting chemokines. They're probably secreting chemokines because they have an antigen, the B cell, they want the B cell to see. So from here, now the B cell is going to get activated and it can turn into a plasma cell, which is basically like an antibody secreting factory, and also a memory cell, which is going to have memory, and then you can remember that antigen in the future. Okay, so we're going to go into this next. This is what B cell activation is next. How you guys doing? Do you guys need a break? I know, there's a lot. There is a lot. Entirely up to you guys. We can keep going. We can power through. All right, let's power through. Unless you guys are like, please, I need a break. All right, let's keep going. So B cell activation and class switching. So this is kind of like the question that someone asked about, why do we have all of the genes for all of the constant chains, right? Like, why do we have all of those if we're only ever putting mu on the surface? And it's for this, it's when you activate the B cell. So B cell activation requires 2 signals. This is like I need to know, because this is going to come up so much with immunodeficiencies again and autoimmunity. So there's 2 signals that must be present in order to activate a B cell. And I remember this because remember like on day 1, I talked about how like the immune system is super, super regulated because you don't want all these cells being activated if you're not trying to activate them. This is why, like, this is one mechanism of like trying to make sure you're not activating a B cell unless you're absolutely certain we want to activate it. So signal 1 is known as the activation signal. So this is going to be, we're going to talk about this, but this is going to be like the actual, like the presentation of antigen. That's what this activation signal is. Signal 2 is known as the co-stimulatory signal. Sometimes people refer to it as the survival signal because if this isn't here, guess what? The B cell dies. And that's why it's like we need both of these signals for the B cell to get fully activated. Co-stimulatory signals are also only present when there's an infection. So again, it's kind of like a foolproof way to make sure that we actually want to activate the B cell. We're not just activating it accidentally. So next we have to talk about the different types of antigens. This is going to become important with vaccines. So the first one are known as thymus-dependent antigens, thymus for T cell. So it's just sometimes people refer to it as T-dependent antigens. So we have T-dependent antigens and T-independent antigens. T-dependent, we have a T cell. So there is a, like this activation requires a T cell to deliver antigen to the B cell to activate it. This is where we're getting class switching. This is where we're getting somatic hypermutation and an affinity maturation. There can also be T-independent antigens, meaning there's no T cell that's helping to activate the B cell. So, but the B cell can still get activated, like it still can get activated by these like low affinity kind of antigens, but it also then releases low affinity IgM. You're never going to get any other class of antibody. You're not going to get class switching. So you cannot get any other class of antibody produced ever. And we'll talk about why the body even, like why do B cells even allow for this? We'll talk about it. But the way more important one is T cell dependent B cell activation. This is like the super strong response. This is the one that we're trying to stimulate with vaccines every single time we get a vaccine. We want T cells to activate B cells. This is the way we get all the different types of antibodies. This is how we get memory. This is how we get like true B cell activation. So this is basically T dependent B cell activation. So here's your B cell receptor, right? So here it's binding to an antigen and it's like, oh my goodness, like I have an antigen that I'm bound to. And again, this is out in the peripheral blood. We're outside. We're like in the secondary lymph tissues now. We're in like the activation stage. So signal one, remember B cells are antigen presenting cells. Remember the three types of antigen presenting cells, B cells, dendritic cells, macrophages. So B cells can chop up this antigen. It can go through MHC2. It can load it on its MHC class 2. Which remember MHC class 2, where is it loaded? Anyone remember? I know it seems like so long ago. Yes. Yes, good. Very good. So this is exogenous antigen and it's going to be loaded then in an endosome. So this is going to get endosomally loaded. Yes, very good. CLIP is going to be involved in this process. It's going to help with the loading. Awesome. Yeah, all those buzzwords, just associate them with your MHC class 2. Perfect. So this antigen gets processed and then it's basically put onto an MHC class 2 molecule. And then it's going to go find a T cell. With T-dependent B cell activation, it's always going to be a CD4 positive T cell. I know we haven't talked about the different types of T cells yet. That's going to be tomorrow. But this is also a T helper cell. So I remember like T helper cell because it's helping B cells get activated, right? So T helper and like CD4 positive, it's the same thing. So this is now helping this B cell get activated. So the T cell receptor is going to bind MHC class 2. Remember the rule of 2, or no, the rule of 8. So CD4 times MHC class 2 equals 8. CD8 times MHC class 1 equals 8. There's your rule of 8. So that's how we remember that CD4 is the one that's binding to the MHC class 2. So T cell receptor with our CD4. This is signal 1. This is your activation signal, okay? But now we need a co-stimulatory signal for this B cell to get activated and to stay alive, basically. Signal 2 for B cells, really important. There's an immunodeficiency where one of like the B cell's lacking this or the T cell's lacking this. So you need to know these. So B cell has CD40 on its surface. The CD4 T cell has CD40 ligand or CD40L on its surface. You do need to know that the ligand is on the T cell and then just normal CD40 is on the B cell. So now we have signal 1, we have signal 2. Great, we can get B cell activation. The T cell, how it's going to help activate it is it's going to release certain cytokines, which is going to tell the B cell, hey, this is what I need. I need IgA antibody. Hey, I need IgG antibody. Hey, I need IgE. So these cytokines they're not like required. That's why it's not like a signal 3. They're not like really required, but they're kind of telling the B cell like it like what it needs basically. Or what it should make, I should say. Okay, so I need to see things, right? So I need to kind of visualize these things. So this is T-dependent activation. So thymus dependent, T cell dependent, whatever. So let's say this is a gram-negative bacteria. Here's a PAMP, LPS on its surface, right? So here's a scary bacteria. Here's my naive B cell. So it's going to have IgM on its surface. I didn't draw IgD, but it also has, IgD on its surface. Here's its MHC class 1 that it has. Here's its CD40. Okay? So it's going to go up to the gram-negative. It's going to bind to it. And it's going to be like, oh, okay, here's an antigen. So it's going to basically process this antigen, and it's going to load it onto an MHC class 2. Because it wants to present the antigen presentation. It's an APC. So it's going to take this down. It's going to take this exogenous pathogen. It's going to load it onto an MHC class 2 in an endosome. And then it's going to basically find a T helper cell, a CD4 positive T helper cell. So your T helper cell has T cell receptor and CD4. That's going to be what's going to be responsible for signal 1. And then in the presence of an infection, we're going to have signal 2 present. So this is going to bind here. So we have signal 1, we have signal 2. Great. The cell is going to survive. Now the T helper cell is going to release cytokines. And again, depending on what type of cytokine it releases, tells this B cell what type of antibodies that it needs to fight off this infection. And again, this is why the T-dependent activation is really, this is why it's very specific and very, very good, because you're getting, this great help from this T cell. So these cytokines, oh, sorry.
Is
I have
this
a
what we
question.
need to, oh, sorry.
Wait, go, Nay.
Go, V, go.
Okay, I'm just confused really quick. So I thought this was going on in class 2. So then what is class 1? What is happening in class 1?
So class 1, I put this on here just to remind you guys that all cells in the body have MHC class 1 on its surface, including the APCs. MHC class 1 really just helps cells determine self from non-self. Remember, one of the things that NK cells do is look for cells that don't have MHC class 1 on its surface, and then it tries to kill it. So I just kind of put that on there as a reminder, but remember MHC class 1 becomes more important with endogenous pathogens, let's say an epithelial cell gets infected by a scary virus. So here's a virus, it infects the cell. The epithelial cell is going to take some of it and load it in the ER onto an MHC class 1 and put it on its surface. And then a CD8 T cell, which we're going to talk about tomorrow, sees it and then kills this cell. If the virus tries to downregulate the MHC class 1 because it thinks it's being slick, then an NK cell will come over here and be like, nope, and then kill it. So you're right, it's not doing anything in this process at all, but I just it was a reminder, don't forget MHC class 1 is on every single cell in the body except, red blood cells. Okay, now you go.
We can use the hot T-bone steak thing for this, right?
So, not really. I'm sorry.
Okay.
Not really. I know. It's just,
Sometimes acronyms and Immuno are just not fantastic.
Yeah, no, that's why that's why it loses its audience. Like I always say, this is really where Immuno loses its audience, is having to memorize these cytokines, and the cytokines have 1,000 functions each. But I'll tell you exactly what you need to know about these. Yeah, you can't use hot T-bone steak for class switching. So we're gonna actually, good segue into class switching. So these you do need to know. You can't use hot T-bone steak. I apologize. If we activate a B cell, the first thing that happens is the release of IgM. Remember, we talked about this. This is why IgM is the first thing that's released in an infection. But then we kind of want to change it and make it more specific to what we're fighting. Are we fighting a parasite? Are we fighting something that would, a virus that would require really good neutralizing antibodies? What do I need here? So this is known as class switching. We're switching from one class to the other. Another name for class switching is isotype switching. Remember, isotype, not idiotype, isotype switching, because isotype and class mean the same thing. And what's a really important concept about, and I'll come back to this, but a really important concept of class switching is we're literally only changing the FC portion. We are not changing the specificity of that antibody. The FAB portion is going to remain exactly the same. Hopefully that makes sense, because we just spent time getting this really specific FAB port, the variable region. So why would we switch that when we're trying to make antibodies against that exact pathogen? So all we're really doing is switching that FC portion to change the class. Again, so that it can go either fight off a parasite, fight a virus, fight a bacteria, what have you. Okay? So there's no change in specificity with class switching. That's why like idiotype switch, it's not called idiotype switching, right? It's isotype switching. So now these are kind of the antibodies you need, like the cytokines you need to know that are important with class switching. They will ask this, guaranteed. If the T helper cell releases IL-4, so like let's say it releases IL-4, this is a signal to the B cell to release IgE. Yes, I have and some IgG because that's just for like, because no one knows anything about Immuno. So it's just kind of to be like, oh yeah, that also happens. But mainly when you get IL-4 release, the main antibody you get, like to class switch from IgM, you get class switching from IgM to IgE. If the T helper cell releases IL-5, then we're going to class switch to IgA. So this is probably more of like your like a GI infection or something at the mucosal barrier. That's when you would want to release IgA. And then here's my rule of thumb. My rule of thumb is if the T cell releases pretty much any other cytokine, you're going to get IgG. That's my rule of thumb. Doesn't hot T-bone steak work? Now I'm forgetting what's 4 and 5 in T-bone steak. Oh yeah, IgE, IgA. Oh, you can. I'm sorry, Nay. I lied. Yes. I did lie. Thanks for calling me out on that. Thank you. No, seriously, like, I forget. Thank you. Yeah, so you can use hot T-bone steak. There you go. One less thing to remember. But anything else, you can get IgG. Does it ever stay IgM? So we're going to talk about that. That's your T independent. So if you don't have a T cell that's helping with this process, it's going to stay IgM. If you mean if like a T cell, no, if you get a T cell that activates a B, like you're going to get IgG. So it'll never be IgM like indefinitely being secreted. And again, this kind of goes back to like when you have an acute infection, IgM is released, but then when we're immune to something, it's always going to be IgG. Always. Okay, so here's kind of like what I just visually, again, really want to emphasize. We're not getting any change to the variable region at all. This is staying exactly the same. The only thing that's changing is we're going, let's say, from mu. So here's mu to gamma. So we're changing from IgM to IgG. So this would be a cytokine other than IL-4 or IL-5 that was released here. Again, really want to emphasize, we are not changing the variable region. That's the whole point of class switching, is we want it to be the exact specificity. We want the exact affinity, specificity. We want all that to be the same. We just want to change the type of antibody it is so that this can go be a neutralizer, go opsonize things, go do what it does. How does the T cell determine what cytokine to release? Yeah, that's kind of like a tomorrow thing. You'll see like because if I go back here, actually, I guess I could have pointed that out. This is nope. Hold on, sorry. Oh, sorry. It is this slide. See how this is an activated T cell? So since it's activated, it kind of knows what it needs from the B cell. So we're going to talk about T cell activation tomorrow. And during that process, it's basically like it figures out what types of cytokines to release to the B cell. But yeah, that's a good question. Okay. Okay, and then this, so the specificity of the antigen from the bacteria is the exact same. We're only changing that constant region. We're only changing the class of antibody. So here's kind of like a little summary. So an antigen presenting cell, so we talked about B cell. So don't worry, actually, we're going to talk about this on tomorrow. Why did I write Tuesday? Oh my God, I'm losing my mind. So we're going to talk about this tomorrow. So Friday session, we're going to talk about like T cell activation. We are going to talk about B7 and CD28, but this is tomorrow. Today, what we talked about was this. We talked about CD40 ligand. So this T cell is going to get activated. This is tomorrow. And then from here, let's say the B cell finds an antigen, it's going to present it on MHC class 2 to the B cell. Here's your signal 1, and then here's your signal 2, your co-stimulatory signal. And then the T helper cell, if this co-stimulatory signal is there, it means it's good to go. It's going to start secreting some cytokines. So it'll secrete IL-4, IL-5, IL anything, and then that'll determine what type of T cell it should, what type of antibodies it should secrete. So the B cell, let's say it was given IL-5, like by the T cell, IL-5. Now the effector cell it's going to turn into is an activated plasma cell that only secretes IgA. If it was given IL-4, primarily it's going to secrete IgE, but remember there's like a little bit of IgG that's released. And then we're not going to talk about this. Well, this is T independent, but we'll talk about that in like a second. But anything else, you're going to get IgG for the most part. Okay. Here was kind of like another just visual of VDJ, kind of like what we talked about. So this is kind of like this is kind of showing right here. This is our VDJ rearrangement right here. So this is our antigen independent diversity development. So V was rearranged, D was rearranged, J was rearranged, and now we have the constant region of mu, which makes sense. This is what's on our mature B cell. Then we get B cell activation. So the B cell is going to get activated. And let's say we need IgE, like the T cell told it that we need IgE. So now we're going to kind of go back through this process and we're going to transcribe the genes for IgE. So over here, we're going to end up with the same VDJ, because again, this is for the variable region. That's going to be exactly the same. But now we're going to have like the constant region is going to be epsilon, which is for IgE. And then everything else gets removed. And this is known as your class switching. Makes sense? So kind of like a little summary. Okay. Okay. So then, I know it just keeps going. So let's say we have a B cell and we had it class switch to produce antibodies that are IgG. Okay, so this is the same B cell that we saw from before, and the T cell told it to class switch to IgG. What it goes through now is fine-tuning. It goes through fine-tuning of that antigen. So we're not changing the diversity. Like, again, we're really not changing the variable region. We're just making it better at recognizing the exact antigen that we just activated the B cell with. And this is what's known as somatic hypermutation and affinity maturation. It kind of tells you in the name. So affinity maturation, we're maturing the affinity of the variable region. And it's somatic hypermutation, meaning like this is not a germline thing. We're not going back to this process and like redoing like germline DNA. We're not doing that. We're basically making these little point mutations in the variable region that make the B cell receptor like really, really, really good at recognizing that particular antigen. And that's just what these terms mean. They're like the same terms basically. So... And you don't have to know the process. No, you don't have to know the process of affinity maturation and somatic hypermutation. You just have to kind of know that it happens. We will talk about it a little bit, but so here's your stem cell. Remember, this is your antigen independent, like generation of diversity. So we have tons of different types of B cell receptors, right? And then they get tested, go through positive selection to make this, then it goes through negative selection. If it passes negative selection, it's allowed to leave. And now it's going to come into contact with antigen out in the peripheral blood, a secondary lymphoid organ, a peripheral organ. And now this is your antigen dependent activation. This is very high affinity. We have antigen specific B cells. And then you're kind of seeing here this process of VDJ rearrangements happening. And then we go kind of like affinity maturation and somatic hypermutation is kind of like, it goes back and forth. I'm going to show you what I mean. I think I show you. Yeah, I'm going to, I don't remember if I show you, but it basically like it tests the antigen, it like sees it, and then it like makes changes to make its receptor better. Then it goes back to the antigen, it like sees it, and then it makes changes to make its receptor better. But again, it's not like a germline thing. It's just kind of doing it. It's like these little point mutations. But it's good. They're good. They're like making the variable region very, very, very specific for that specific antigen. Okay, so then after we get somatic hypermutation and affinity maturation, we have like this is the B cell, these are the receptors that are very specific for that antigen. Now we want to remember, we want to both remember that antigen in the future. So if we see the same LPS bacteria again, we want to remember it immediately so we don't have to go through this long tedious process again. And we want to get plasma cells made. And plasma cells, remember, these are your antibody secreting factories. So they're just secreting tons of whatever antibody the T cell just told it to make. And I have here too, like this is the exact one we just made after that antigen dependent activation. So both somatic hypermutation, affinity maturation, and class switching. So whatever the last final antibody is, after it goes through all that fine tuning and everything, then the plasma cells are gonna start secreting it. And remember, plasma cells, these are the ones that are in those medullary cords. So remember, we talked about anatomy with anatomy on Monday. Plasma cells can be found in the medullary cords. Remember, I said, like, I remember that because it's like as the B cells like leaving, it gets closer to basically the medulla, and the plasma cell is like a more mature B cell, basically. And then we have memory cells, which look exactly like the B cell we have now. So if we ever encounter the antigen again in the future, it'll just immediately get rid of it. But you really only get memory cells and plasma cells in T-dependent activation. And that's why T cells are really, really, really critical for B cell activation. So that's T-dependent. T-independent is like very quick. It's basically like, how do you get a B cell get activated without a T cell? And this is kind of back to that question, like, when do you ever get IgM? Like, when do you ever just get IgM getting released? This is when you only get IgM getting released. And we'll talk about that. So this is obviously a weak response because the T cell's not kind of telling the B cell what it needs. But this is me saying, like, I think the body's like, oh, whatever, it's better than nothing. You know, IgM can fix complement, right? Like, IgM's really, really good at activating the complement system. So, like, at least you got that. So I think that's why this process happens. So this is like a really good picture of T cell independence, so thymus independent, T independent, whatever, B cell activation. So here's your B cell. It's going to recognize some sort of pathogen and it's just going to kind of bind to it, but it's not going to be able to make a really, really strong response to it. So what you get is you just get pentameric IgM getting secreted. That's it. You don't get class switching. Oh yeah, you only get IgM. It's weak affinity. It's weak affinity IgM, so it's not even going to be that strong against this, but it's still IgM. So this is what I'm like saying, like, oh, you're still going to get complement activation at least, right? But yeah, you don't go through somatic hypermutation, you don't go through affinity maturation, you don't go through class switching, you don't make any memory B cells, and you don't make any plasma cells. This is all you get, just secretion of pentameric IgM. And a lot of the times too, this is what happens with polysaccharide antigens. Do you guys remember? Probably not. But on Tuesday, I have like a slide and I say that T cells can only recognize peptides. That's it. T cells can only recognize peptides. So polysaccharides are not peptides. So that's why a T cell can't take this and break it down and get activated from this antigen. So the B cell's kind of on its own. It's like, all right, well, I'll try. It's like, I'll try to fight it off. So that's why like it at least will secrete some IgM, which is better than nothing. And again, and then we'll talk about this with vaccines, but if you want to make, like, polysaccharides are what make up capsules, right? So if we want strep pneumo, like some people get strep pneumonia vaccines, right? Like I just told you, if you don't have a spleen, you should get a strep pneumo vaccine, you should get a Haemophilus influenza vaccine, you should get a what's the other one? Neisseria meningitidis vaccine against the polysaccharide capsule. And we'll talk about this, but what they usually do when they're developing these vaccines, they couple a protein to it so that T cells can at least see it. And then you get a much stronger response than if I just injected the polysaccharide capsule, I would only get this pathway. Does this pathway happen along with T cell dependent? Why would you ever want a weak response? You don't want a weak response, but T cells can't see a pathogen like this. It can't break down polysaccharides. T cells can only load peptides into its not load. It can only be activated by peptide antigens, T cells. So a polysaccharide antigen, which is what it's saying here, so this is like the only possible way to get rid of something like this, is this T-independent B cell activation pathway. which is not great, but it's better than nothing, right? So it's like, all right, it's better than nothing. At least I'll get some complement activation, not a big deal. But like, this is why, and I'm going to come back to this when we talk about vaccines, you usually couple these types of antigens to peptides so the T cell can see it. So you get the T-dependent B cell activation. So here's kind of a summary of what we just talked about. So T independent, again, it's going to be like a polysaccharide. It's going to be something that's just going to kind of cross-link, and I should have pointed it out here. It does cross-link the B cell receptor, so it has to be big enough to do that. Like if you just have like a little polysaccharide like this, you're not going to get B cell activation. It has to at least cross-link some of the B cell receptors. So like this is another example. A lot of the time it is a polysaccharide, so it is like a polysaccharide that, because they're really long and they can kind of cross-link all the B cell receptors. But you can't go through, like the T cell is not going to be involved at all because it doesn't see this antigen at all. So you get low affinity IgM. You get an immune response, but it's weak. But again, you're getting complement, so it's better than nothing, right? T dependent, this is a lot better. And I say here again, T cells can only see antigen in the context of MHC. MHC can only load peptides. So that's why you kind of need peptides if you want a T dependent response. So this image isn't great. It's not showing signal 2. Remember, you need signal 2. So this is showing signal 1. We need signal 2. We need co-stimulation. So we need CD40 and CD40 ligand. Okay. And then the T cell, once we have signal 2, then the T cell is going to secrete cytokines. So then you're going to get very high affinity IgG, IgA, IgE, depending on what it's secreting. But it's going to be a very, very strong response. And this is what I'm talking about. If you couple a polysaccharide vaccine to some sort of peptide fragment, a lot of the time they use like a diphtheria toxoid, or they'll use like some sort of like toxin that like diphtheria, pertussis, tetanus, like one of those toxins. They just take a portion of it and then they couple it to the vaccine, to the polysaccharide, and then they give that. That's what they're injecting in you. All right, so here's another summary. So this is an overall summary, but again, it's all the same thing we kind of talked about. First, we get antigen independent, like development of diversity. So we go through DJ rearrangement, positive selection, VDJ rearrangement, positive selection. Then we're going to get, it's not shown, but we're going to get the light chain rearrangements and then positive selection to give us our final like mRNA. And then from here, it's going to go stem cell, well, this isn't all inclusive, but whatever. So then it goes to like a secondary lymphoid tissue. So this is now antigen dependent. So it's going to come into contact with some sort of antigen, and then it goes through somatic hypermutation, class switching. It's going to do this until it finds like the perfect match. And then you're going to get release of your plasma cells and memory cells. And of course, this is only with T dependent. And then this is kind of saying, I don't know why it shows it all the way here, but somatic hypermutation in the germinal center. This is over here. This is out in the secondary lymphoid tissue. Okay. So again, I kind of brought back up this concept of central tolerance and peripheral tolerance. Today in my lecture for Immuno, I really liked this slide, so I stole it. So don't tell Dr. Mueller-Weeks, but no, I'm kidding. But I took this slide from her presentation today because I really like it. It's basically like another summary, right? So it's like another summary of everything we just talked about. So we're in the primary organ. So we're in like the bone marrow here. And central tolerance is like this idea of like not passing these, like if you have a self-reactive B cell, you either delete it or you undergo receptor editing. And remember, receptor editing, you have to upregulate the RAG genes again. And then from here, or you just have no self-reactive B cells, they're allowed to leave too. So they're going to pass negative selection. They pass. So they're going to make it through and leave. Literally, so they leave. And then when they're out here and they come into contact now with foreign antigen, and they have a co-stimulatory signal from like probably a CD4 T cell, so this is probably a T cell here, then you're going to get production of effector and memory cells. Both of these is showing that. But let's say it's now reacting with self-antigen, or it's reacting with foreign antigen without a co-stimulatory signal. Then you can either delete it, so apoptosis. You can inactivate it. Remember, the other word for inactivation is anergy. So it's like still there, but it just doesn't do anything. That's anergic. We didn't talk about clonal suppression, but it's just like another, you don't have to worry about it. It's just kind of like it's not able to work. But all of these lead to what's known as peripheral tolerance. And we're going to go into a lot more detail with like central tolerance and peripheral tolerance with T cells, because again, it's really important with T cells, but just know that B cells do go through this process. We did kind of talk about it, but we're going to go into more detail tomorrow with T cell development. It goes through the same process, basically. We did it. We made it. I did warn you. I did warn you guys that this is how it was going to be. You guys have any questions? We're going to run through some of these. It's up to you how many you guys want to run. Yeah, B cells are wild. B cells are incredible. I can't wait though for you guys to see T cells. T cells are even more wild. I love a good T cell. So tomorrow we'll talk all about T cells. Questions? You want to do some questions? I just kind of want to show you how they would ask questions about this insane process. We can take 5 minutes. Absolutely. Yes, I didn't give you guys the break at 7. Let's take 5. I'm going to pause the recording. So if anyone has any questions, I'll sit here. Pause. What would I do without you? Thank you. All right, recording people. This is the first question. Thank you, Alex. Okay, that was about 1.5 minutes. I know it's going to be quick because I know this is like, we just talked about it. But so I think a decent amount of you guys have answered. So 2 vaccines meningitis has a capsule, right? So 2 vaccines for meningitis. One is just the polysaccharide. One has this toxoid. So this is going to be B. So very good if you guys, very good. Again, I know it's like a lot to kind of take in. So if you didn't get it, not a big deal at all. Now you'll get it for the next time. It's not, so like, let's talk about why the other ones are wrong. So it contains amino acids. It's just another way to say it's a protein, right? So the diphtheria toxoid is a protein. So the first vaccine cannot elicit a B cell response. That's not true. It can. It's just very weak. It's just that IgM only release one, right? So it's just a very weak B cell response, but you still get one. The second vaccine activates complement. Both vaccines activate complement, right? We're still going to get IgM in both instances. If this said the first one, that would be correct, but also that wouldn't be the answer to the question. Why does the second elicit more of a vigorous immune response? So that's out. First vaccine elicits a T cell dependent response. No, we talked about you can only, T cell dependent is only with proteins. And then second vaccine is presented by MHC class 1. No, it's still going to be presented by class 2, but again, you need an activated T cell to help with it. Good? So it's kind of that thing we talked about like at the end. Here's this. So it'll be, again, I'll send it out there. This could have also, instead of saying diphtheria toxoid, yeah, it could have also been anything in like the D tab. So pertussis, diphtheria, or tetanus, all of those are commonly used as like toxoids to be coupled to polysaccharide vaccines. Okay, next question. This is, read this question carefully. I almost got on this, just doing it right now. Yeah, I know, this is a tough one. And I love that you guys keep coming in with all these answers. We're having a good time talking about this one. A lot of you guys are getting gut. I'm just gonna say. I'm just gonna say. Okay, let's talk about this one because we're all over the place. But this is a tough question. Okay. So basically what they're saying is this woman who's pregnant is recruited for this clinical trial. She's administered a vaccine. Okay, so serum testing indicates antibody response to group T streptococcus polysaccharide capsule antigens are present in the vaccine. So now she gives birth to a baby. After the baby's born, the baby develops irritability, lethargy, and tachypnea, which is signs of infection of group B strep. So then it says, which of the following most likely describes a class of antibody generated in the mom in response to vaccination? So some of you guys picked IgG for this. Actually, most of you guys picked IgG. You're the ones that got. Why can't it be IgG? Some of you guys said it.
Is it because like that one isn't the one that participates in complement?
IgG does participate in complement.
I meant like the oh my gosh. I don't know what I'm trying to say. It's like it doesn't have a bunch of it's not it has a bunch of antibodies around it.
It's not the IgM, right?
Yeah.
The
Yeah.
multivalent one.
Exactly. Yeah, that.
So the trick with this question was this part right here. So the baby wasn't protected against group B strep. So mom got vaccinated, but it did nothing for the baby. So what does that tell you? Mom didn't make what antibody that did not cross the placenta?
IgG.
Mm-hmm. That was the key here. So you guys answering IgG, you're right. Like that's what should have happened. She should have, but this polysaccharide capsule, she probably didn't even generate IgG. It probably wasn't coupled to a like a protein. So she probably only made IgM. Oh, I gave away the answer. All right, she probably only made IgM because she just was a polysaccharide. Now, if it said, some of you guys mentioned class switching, which it's tricky because you can technically, it depends on if the T cells were activated, which we haven't talked about yet. So this wasn't really like supposed to get you thinking about class switching. It was more so like this, like the baby was not protected against the infection. The baby, oh, it's a boy. He could not have gotten any IgG. If mom made IgG and the boy was born and he was healthy, then probably more likely than not, this polysaccharide capsule was coupled to some sort of toxin, so mom was able to make IgG. She was able to class switch from IgM to IgG. Does that make sense? This was a tricky one. I know, it was a tricky one. Okay, good. And this is kind of what it's mentioning here. I really think the key was like seeing that the baby was not protected. And yes, some of you guys did mention IgA. We're going to talk about this when we talk about vaccines. You really only get IgA to a vaccine, which like how would you have known this, right? Is if you have like an oral vaccine. So something like one of the there's two types of polio vaccines. One was Sabin's and one was Salk's, Jonas Salk's. One was given IM, one was given orally. The oral one, that's when you produce IgA. There's a type of flu vaccine. One of the types of flu vaccines you can get intranasally, you would make IgA. But you really don't make IgA from vaccines. It's either only IgM or IgG. Does that make sense? And again, it's just kind of like, how would you have known that? We haven't talked about that yet. But it's just like a thing with vaccines. Like, you can't get IgA from a vaccine. Okay. Can we go back to the question? Yeah, sorry. Let me cross this stuff off. How did the baby get sick? So this is another thing you probably guys don't know, but like, if you're a pregnant mom, so if you're pregnant, all pregnant moms are given antibiotics against group B strep, like regardless, they're given this antibiotic against group B strep right before delivery, because group B strep is found like in the vaginal canal. It's just, it's completely normal. It's like a commensal organism. It's not bad, but it can infect the baby. So regardless of anything, pregnant, like right before you deliver, you're given an antibiotic for group B strep. I think it says it in here. It might. Oh yeah, right here. GBS, yeah. So mothers colonized with GBS receive antibiotics at the time of delivery. So it's like they test you for a group B strep. Again, it's normal. It's not like the mom's going to be sick. She's not going to be sick at all. It's just like they test the mom's vaginal canal, and it's like, let's see if there's group B strep in here. And if there is, they're given an antibiotic because it can infect the baby. So this was kind of saying like, all right, mom probably had group B strep. She probably did. They didn't tell us, but she probably did. I mean, she obviously did. The baby got group B strep. But instead of giving her an antibiotic, they gave her this vaccine like a month before. So they knew the baby would get it. Well, this probably wouldn't be allowed, but the question writer knew the baby would get it, I guess. Yeah, I know. It's messed up. I know. All right, but does that make sense? So this is kind of like it's, again, there's a lot of stuff in here that you guys didn't really, like, shouldn't know yet. But it was just kind of getting at, like, do you understand, like, if the baby has symptoms, that means that the baby did not get protection via mom's IgG, which is the only one that can cross the placenta. So that means mom must have only made IgM. Okay, cool. Awesome. All right. Next one. Okay, I'm gonna go to the next one. I'm sorry if you guys didn't answer. I want to get to other ones, but I wanted to show you that they will ask. They will ask about portions of the antibody, and I didn't. I'm so proud of you guys. No one got. Thank you for no one picking this one. But this one is gonna be C. Don't get got. Idiotype is not isotype. Yep, the old fixing of complement. Exactly. It's always the answer, right? Yeah, so I just wanted to remind you guys too, like the complement binding domain is it's down here in the constant region. And actually this is also, they could have had where macrophages bind, right? So like where macrophage like receptor, like FC receptors, FC gamma receptors bind to. So that could have also been here. But yeah, idiotype is for the FAB. Idiotypes are FAB. I guess, yeah, FAB. Yeah, all right. And then isotypes are going to be, so this would have been correct if it was isotype, basically. Cool, okay. Very good. And I put this on here so you guys wouldn't get got. All right. All right, next one. Don't get too caught in the details. We didn't talk about a lot of this stuff. Yeah, this one, if you guys went back and looked, I wouldn't blame you. But I just wanted to show you that they will ask about characteristics of all these immature cells as we talked about, or mature, immature, or mature. Okay, so let's talk about this one unless anyone really wants to get their last answer in. I'm going a little quick, but there's a lot of questions I want to kind of get to. Okay, so this one, the key here, blah, 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 cytoplasmic mu heavy chains. One of them, I even highlighted on the chart, one of them has cytoplasmic mu, remember, and then it goes up to the surface mu. Surface mu is going to be your immature B cell. So a lot of you guys picked A. You're close, but it's surface mu. So cytoplasmic mu is your pre-B cell. So your pre-B cell has cytoplasmic mu. Pro is before pre. I think some of you guys got confused there because a lot of you guys actually also picked pro. So it goes pro, then pre. It's like large, then small, and then immature, immature. So like this order would be like B, E, like of development. B, E, D, A, C. And then remember, it's like early and late pro B, large and small pre, nope, large and small pre B. Oh, I did not hear. Early and late, large and small. And this is surface mu, cytoplasmic mu. That was really the only way to kind of do this, and that's what they're saying here. Don't worry about, we didn't talk about a lot of these antigens. You'll get a lot of this with hemonc, in hemonc block. You'll get a lot of these, like knowing all the CD markers for all the cancers. It's really fun. Okay. Okay, next one. Okay, a lot of you guys are asking me questions. Okay, so let's go through this one. I also love that, like the classic, oh, it was a lab error. I love that. No, it's unfortunately not a lab error. All right, so let's talk about this. So male infant, okay, so no prenatal care. That doesn't matter, whatever. Mom seems fine, that's great. Baby clearly has some issues going on. She's got hepatosplenomegaly, spleen's enlarged, liver's enlarged, microcephaly, head's tiny. You didn't have to know that this was CMV. This is how CMV presents, but you don't even have to know that. The antibodies tell you what's going on. So mom, she has IgG. Oh my God. And has no IgM. What does this tell you about mom? So has IgG, but no IgM. Is mom actively infected? Good. So she had it before or she was vaccinated. That's pretty much you can't tell with positive IgG. So either vax or she recovered from infection. Baby, hepatosplenomegaly. There's some crazy words in medicine, I'm telling you. Hepatosplenomegaly is a good one, though. All right. Baby has IgG and has IgM. What does this tell you? Actually, what is the positive IgM? What is IgM? The baby having IgM, what does that tell you?
The baby is having an active infection.
Yes, good. But then you're sitting here and you're like, well, why does she have IgG then? Like, shouldn't she be recovered? But who has IgG? Mama. So mom passed her IgG over to the baby, but guess it wasn't enough because baby still's got active infection. So this one is congenital infection with CMV. So the baby has CMV like actively right now. That's what IgM tells you. Like no ifs, ands, or buts about it. If you have IgM, you are having an infection and you have like these signs and symptoms of an infection. But the trick with this one was like, well, where did the positive IgG, like where is that come? This should mean that the baby's vaxxed or recovered, but it could also mean it crossed the placenta. It came from mom. So that's what this is, like passive antibody transmission from mom to baby. This is the IgG portion, but this describes the baby's like symptoms basically. Make sense? And this is like a really good, again, a really good like diagnostic tool with testing. Like if you look to see if someone has IgM and they have IgM, they have an active infection. It's always going to be the case. Okay. Good. No last questions on that one. That's like a really, really important thing I really wanted you guys to take away. I know this is, I feel like I shouldn't have put this on here yet. There's a lot of stuff we haven't talked about yet, but just try your best. You guys ready for this one? You're starting to ask me questions, which I always can tell. Which is good, because you want to talk about it. Yeah, so a lot of you guys got what this was. No, don't take it back. You were right. Don't take it back. Yeah, it's good. So a lot of you guys actually got what this was, which you didn't have to know what this was. But this is Giardia. And remember what I said, what antibody you need with Giardia? IgA, right? So this is exactly how Giardia presents. They're hikers, they're people whitewater rafting, they're doing something and they drink the water out in the streams. Like that's how you get Giardia. So you need IgA to protect against Giardia. But this is saying secretion of these antibodies, IgA, most likely requires binding of which of the following. So a lot of you guys did kind of pick up on this. It's saying like which one of these is one of those signals that's absolutely required to even get to class switching from IgM to IgA. So this one's going to be B. This is your co-stimulatory signal for B cells. Yeah. Yeah, you guys, I was so proud. A lot of you guys, I'm proud. And some of you guys picked this one. This is not like, it's not wrong because this is like a co-stimulatory for T cells, which we're going to talk about tomorrow. But it's not like, that's not how you would get class switching though. This is just how you would activate a T cell. So like if you guys knew what these were, this would have probably been a harder question. Like, because you would have been like, well, how do I know which one it is between these two? This is the co-stimulatory signal for T cell activation, but we're talking about class switching, which is a B cell activation thing. So we need the co-stimulatory for B cells, which is CD40 ligand. And which one of these two is on the T cell? Yes. Good. Just L. Very good. Yes. So, and that's something important to know because again, with the immunodeficiency, it's a CD40 ligand deficiency. We're going to talk about it. So you can imagine it's a T cell issue. So we'll talk about it when we get to immunodeficiencies, but just keep this in mind. So this is B cell activation, co-stimulatory. Tomorrow, we're going to talk about this a lot. And this is your T cell activation, co-stimulatory. Okay. Okay. Cool. So what it says here, here's kind of like another picture, which is kind of cool. But this is like showing your, this is basically showing your B cell activation, like T-dependent B cell activation. Okay. And yeah, so that's what it also says here. Like if this said like if this was CD4 to MHC class 2, that could have also been a correct answer because that's your signal 1, right? This is signal 2, but both are required to get class switching. To get class switching to IgA, which is really what you needed here against Giardia. Okay, next. This is a throwback question. See if you guys remember stuff from Tuesday. Sorry. Okay, last answers. This was like one of those like strict recall type things. It's okay if you don't recall it right away. It's like day 4 of Immuno. Okay, so this one, neutrophil infiltrate. So an innate thing that calls in neutrophils, basically. So this is going to be IL-8. Some of you guys clean up on IL-8. Remember, clean up on IL-8. Clean up for your macrophages, or sorry, oh my God, your neutrophils. They're cleaning up on IL-8, like IL. And nay, I like yours better. 8 for 8, because neutrophils are phagocytic. I actually really like that. That's really good. Yeah, some of you guys did, I think, jump to this one because maybe you thought this was 5B. That would have also been correct, but it's not. It's 3B. And 3B is your opsonin, right? IgG, it doesn't really attract neutrophils can bind to it. They have Fc gamma receptors, but it doesn't matter. Myeloperoxidase is not relevant here. And TNF alpha, it does induce an immune response. It will do that. It's just this is the sole purpose of IL-8 is to call in neutrophils to the area. That's kind of what it says. I'm wondering if it says clean up on IL-8. No, it doesn't. Okay. Next. For bonus points, you can tell me what antibody this is. Oh, I know. It's been a long day. I know. I'm aware. I know. And like I say, I give you guys these questions just to see like everything we talked about is relevant. Like I promise there's boards questions on all of the stuff we talked about. So that's why I like to do questions. Okay, let's go over this one. So, because I know you guys are tired. So this one, a lot of you guys got the antibody, which is great. So, produce an antibody that prevents attachment of pathogens to mucous membranes, but does not fix complement. So this is IgA. So remember, IgA exists as a dimer like this. And since the FC region is kind of like hidden with this J chain here, complement can't bind here. It can't get fixed. It can't get bound. It can't bind. So it doesn't activate complement, and this is the guy that protects against mucous membranes. So then, yes, hot T-bone steak. So hot T-bone steak, it's the A in steak. So this is IL-5. Okay. And then, yeah, IL-4 is going to be for IgE and IgG. Okay. There's this one and then 2 more questions. Wow, not as many of you guys are getting got. I am very proud. I'm very proud. But we've also been talking for a very long time, so if you're getting got, it's fine. Okay, you guys ready? Chat about this one? Okay. All right, so my people that got. Yes, I know it's a parasite. Yes, I know how badly we want to pick IgE because it's a parasite. This is not the answer. I'm sorry. I'm sorry, but I'm pointing out things, common ways that people get got. Acute infection. What is the best way to diagnose acute infection? This will only ever be IgM. It will always be IgM. Yes, good. Yes. So yes, I know it's parasite. Yes, it's a parasite. Yes, it doesn't matter. If you have IgE, it doesn't matter. Acute infection, IgM. Okay? Just start like, and this was another tricky one of how they can try and be like, oh well, maybe it's IgE because it's a parasite. Doesn't matter. Acute infection, IgM.
Christina, wait. Okay.
Yes.
So, I'm confused because I thought it's a neat, like, they're not. Oh, never mind. I thought the baby was in the, like, womb still. I didn't realize it was born.
Yes. Yes. Yeah, good
In
question.
kids, if they asked about, like, a person, like, a baby that's not, like, that's in the womb, essentially, they only have IgG because it
Yeah,
crosses?
babies don't like in the womb, you don't produce
Yeah, okay.
antibodies. And even so someone even just chatted this to me. So babies up until about like 6 months, the only antibody they can even produce is IgM. That's it. That's why like babies are considered immunocompromised. And that's why babies though can survive with mom's IgG for like up to 6 like that's what's protecting the baby for the first 6 months of life basically.
So if you want to test for like certain autoimmune disorders, do you have to wait until like after 6 months?
That's why a lot of the times too, like some babies are born completely normal. And then it's like after, and we'll talk about this with some of the immunodeficiencies next week. but it's like presentation is like year one, and you're like, why? Like, this is a genetic defect. Like, shouldn't they have been born with it? And it's because you have no idea, because they're not producing antibodies until about 6 months to a year. Yeah, but those are good questions. Yeah. So again, yes, it's still, it is, I know you could have kind of figured that out then if you knew that babies only produce IgM, but the takeaway, acute infection, that they're getting at IgM. They want you to pick IgM for acute, like, active infection. Just like that, like one of those first questions we did where like baby had positive IgG and IgM. That's an active acute infection, even though she also had IgG. She just got that from mom. Let me see if it even mentions it here. Okay, and then even here, this says IgE doesn't get produced until the second year, which I guess it's kind of, you don't have to memorize when they start producing certain antibodies, but it's like not, it's definitely not right away. It's definitely at least like 6 months to a year after. Okay. All right, second to last one. You guys are gonna hear me like during your test, like, don't get got, don't get got. Okay, it's about a minute and a half. Any final guesses? No, you guys did not get got. Good. Okay, so this all is asking, where does VDJ rearrangement occur? That's pretty much it. Bone marrow. Remember, what you need, it can only happen in the bone marrow. It is a genetic thing. It is DNA like rearrangement that can only happen before it leaves and goes to the periphery. Okay, does that make sense? Good. And like the RAG gene, nice little reference with the RAG gene mutation, right? So you knew, even if they didn't tell you VDJ rearrangement was involved, if you saw a RAG gene mutation, you knew VDJ rearrangement was going to be involved. And again, this is a congenital immunodeficiency syndrome we will be talking about where you have a RAG gene mutation. So it's good you're starting to realize the implications of that before we even talk about it. Okay, cool. So VDJ happens here. Okay. And then here's kind of, this was like Amboss's picture of VDJ. I just thought it was not confusing, but it's like, wait, what? What's happening? But I wanted you to kind of see it again, just kind of like see the process again, all the way down to the heavy chain and then to the light chain, but light chain's only VJ. Okay, last question. Yeah, when you guys are waiting too, try to see if you can list all the antibodies these are referring to. It's good practice. No, you don't have to send it to me. Just just in your heads. I think a lot of you guys answered. Anyone holding out? Just want to get it over with. Send me something. Okay. All right. So this one, the kicker here, mucosal associated lymphatic tissue, mucosa. So what antibody are we looking for?
IgA.
Yay, very good. Good. So now this is a classic step question. It's like you know the answer, but that's not what they want. They want that step above. So now you have to know what IgA looks like. So IgA is a dimeric immunoglobulin with secretory component. Remember, secretory component is it grabs it from transcytosis, the poly Ig receptor. It kind of grabs part of it, and that becomes your secretory component. What is B? A monomeric immunoglobulin that crosses the placenta. Yes, good.
GG.
Yes, amazing. How about a monomeric immunoglobulin bound by mast cells?
Is that E?
Yeah, very good. We're going to talk, I know I realized I didn't really talk about this too much. When we talk about type 1 hypersensitivity, I go into like a lot of detail about it, so I didn't want to like do it twice because you'll be like, all right, we heard this already. But yeah, IgE is always just bound to the surface of mast cells, just always. Okay. How about D? Monomeric immunoglobulin that opsonizes.
Is this IgM?
No.
I think it's IgG.
Nay, the reason it's not IgM is because IgM, it's not really an opsonizer because when it's floating around, it's a pentamer, which I kind of gave it away, but it's a pentamer. And it's not good at opsonizing, it's good at activating complement. But remember, opsonization itself is like, here's a bacteria, and now I'm going to coat it with like with antibodies so that cells that have FC receptors can like see it and eat it, basically. So usually opsonizers are your monomeric. And of course IgG is like the best opsonizer of the antibodies. Actually, I think it's the only opsonizer of the antibodies. And then, yeah, so E, this would be IgM. That's like what IgM does. So when IgM is monomeric, it's just on the surface of B cells. It's not like it's floating around doing stuff. And then IgG is not here because, again, no one knows what IgG does. But in theory, it could have been like a monomeric immunoglobulin found on the surface of mature B cells. That's probably the only thing I would think that they would say about IgD. Okay, good. All right. Stop recording.